All right, welcome everybody. Today is our August webinar. It's our great pleasure to have a guest speaker, Dr. Ruan, with us today. My name is Zhiying Li. I'm the Director of Life Science and Technology at GEM Pharmatech US. And today is my great pleasure sharing this stage with Dr. Ruan, speaking at the portion where we provide the webinar from our headquarters in China. So before we get started, I have a brief introduction of Dr. Duan. I'll share with you. Dr. Ruan is Associate Professor in the Department of Integrative Biology and Physiology at University of Minnesota. He's also a faculty in microbiology, immunology, and cancer biology. He has a broad scientific background and interest in genetics, metabolism, immunology, and physiology. Haibin received his PhD from Nanjing University. His thesis work was mentored by Dr. Xiang Gao, the founder of GEM Pharmatech, as many of you know. His work during this time culminated in a high-profile publication in Nature Communications, researching on the role of stem cell factor KIT signaling in myeloproliferative disease and energy metabolism. Haibin moved on to his postdoctoral research at Yale University. His work established the essential function of O-GlcNAc as a nutrient sensor in glucagon-induced autophagy, hepatic glucose metabolism, and neuronal control of beige adipocyte function. After establishing his independent laboratory at University of Minnesota in 2016, Haibin has been focusing his research activities on understanding how environmental cues and intrinsic signals are joining force to regulate metabolic processes in health and disease. He's taking an integrated approach in studying tissue adaptation and remodeling upon metabolic stress in 4 major areas: O-GlcNAcylation in physiology and disease, adipose remodeling and energy balance, in intestinal epithelium and gut microbiome, and finally, immune homeostasis and systemic metabolism. These studies have broad implication in obesity, diabetes, inflammation, and aging. So today is a great pleasure to hear Dr. Ruan on these topics, and in particular, he will be talking about O-GlcNAcylation and its function at the crossroad of immunity and metabolism. So without further ado, Haibin, the floor is yours. Thank you so much.
All right, great. Can you hear me okay? All right, thank you so much, Jim, for the invitation and also the kind introduction. It's my great pleasure here today to share a couple of the stories that we recently has been doing in the lab. So before I get started, I just one quick correction that I'm not affiliated with Department of Immunology. I'm actually just associated with Center for Immunology. So today we're gonna, okay, my slides are not proceeding. Okay, just one second. Oh, sorry about that. Okay, so as Jean just mentioned that we are interested in cell-cell communication in metabolic and immune systems. There's a few directions right now ongoing in the lab. When I was trained in Dr. Xiang Gao's lab, I've been studying metabolism, so I have long-term interest in that. So I'm still working on that. It has a lot of effort at working on adiposity remodeling, obesity, and aging. But I also have a long interest in immunity. So since I established our own lab about 6 years ago, we got more and more involved, really studying not only just the protein oxidation, but also other metabolic pathways in immune systems. Particularly, we like to use intestine as a really great model to investigate host and microbe interactions. Okay, so as the title indicated, there will be a sweet component in this in today's story. So when you talk about glycosylation, I guess most of the audience is going to be thinking of these structures that happens on intracellular proteins as listed here. On your left it will be these O-linked glycosylation, on the right will be these so-called N-glycanes. As you can see from here, these extracellular glycosylations or really happens on serine, threonine, or asparagine sites that can happens at branched and complex structures. If you notice that these different color-coded square or circles actually or derivatives of glucose metabolism. For example, this blue square, my favorite, is actually called N-acetylglucosamine or O-GlcNAc, is involved in most of the glycosylation structures. So, as the name suggested, it would be a sweet modification. For those who in the audience right now maybe thinking or maybe shouted out and that, oh crap, I'm gonna hear these boring structures for the next hour. I have good news for you. So I will not actually talk at all about all these structures. Instead, we're gonna talk about these more simple modifications of the intracellular proteins, which is called O-GlcNAcylation. So what is this modification? This is actually an O-linked N-acetylglucosamine happening on the serine and threonine residues of intracellular proteins. Interesting enough, there's only one pair of proteins that are modular these modifications. That is these so-called O-GlcNAc transferase or OGT, adding the sugar moiety to the protein. On the other hand, this O-GlcNAcase or OGA, removing the sugar from the protein. So this protein, compared to the aforementioned traditional N or O-GlcNAcylation, in three major aspects. First, it is only a single O-GlcNAc moiety. There's no complex or secondary modifications. Secondly, it happens almost exclusively in intracellular proteins. And also, this is, as you could see here, it's actually a dynamic process. It's on and off can happen very quickly and in response of different physiological, pathological stimuli. So a lot of labs in the world, including ours, are interested in this modification, largely because the substrate for this modification, called UDP-GlcNAc here, is actually being considered as a nutrient-sensitive pathway. As you can see over here, the synthesis of UDP-GlcNAc utilize a so-called pathway, hexosamine by synthetic pathway inside of the cell. When the glucose entering cell, besides to be glycosylated, entering glycolysis for and also for glycogen synthesis, a portion of the glucose actually can enter in this hexosamine bisynthetic pathway and utilize glutamine, acetyl-CoA, and also UDP to generate this UDP-GlcNAc. So as you can imagine, the availability of the substrate is really heavily dependent on the availability of these nutrients that I mentioned on the left. So since its discovery by Jerry Hart in 1980s, there's growing numbers really working on not just the O-GlcNAc proteins, but also the size and the functions in different molecular and physiological conditions. But we still, this O-GlcNAc is still relatively small field. As you can see that there's only about 200 papers a year. But there's a growing interest in a scientific field in terms of the physiological importance of these modifications. As you can see on the bottom right, that there are more than 10,000 proteins has been identified to be O-GlcNAc modified. If you're interested, I encourage you to go to this O-GlcNAc database and then find whether your protein of interest is O-GlcNAc modified or not. So the studies over the years have really focused on this O-GlcNAcylation, as I just said, as a nutrient sensor to regulate physiology. However, since my postdoc work and continuing in my current lab, we found that O-GlcNAcylation is not just a nutrient sensor. We found that a lot of stress, including nutrient stress, for example, starvation, oxidative stress, can lead to increase of glycosylation. Our colleagues at Yale and many other labs have shown that circulation is another important regulator of O-GlcNAcylation in metabolic tissues. And recently we found that microbiota is also important in terms of maintaining the O-GlcNAcylation levels, at least in the intestine tissues. So the overall aim of my lab is really to try to understand, when O-GlcNAcylation is activated and what are the role of this O-GlcNAc modification inside the different cells, particularly metabolic and immune systems. I just want to mention that it is very in terms of function, it is also very like phosphorylation. Its activity is really context and protein specific, depending on the stimuli, depending on the cell types, specific cell types you study, and depending on the protein that it be modified. This O-GlcNAc modification can have a diverse activity, for example, affecting cell-cell interaction, protein-protein interaction, affecting protein stability, and also can affecting enzymatic activity as well. As I just alluded earlier, that my lab is really interested in really study microbe-host interactions. For many of you in the audience probably knowing that those so-called molecular microbial pathogen-associated molecular patterns are important for the host to recognize as a foreign or non-self subject, and hence to activate the immune systems. These so-called pattern recognition receptors are so important to inform the immune systems there's an infection. But at the same time, those pathogens, no matter it's bacteria or virus, they also always a lot of times hijacking the host cell for their infection, and a lot of times these are through post-translational modifications, for example, glycosylation, ubiquitination, and many others. So when I studied the lab 6 years ago, almost 7 years ago now, we simply ask ourselves whether O-GlcNAcylation is also important in microbe-host interactions. As a matter of fact, this has been really studied a long time ago, not long after the discovery of this O-GlcNAc modification. People has been found that the T cells, that when they are activated, you could observe a transient but robust activation of protein O-GlcNAcylation inside the T cells, particularly in the nuclear fraction. And not only the modification, but also the substrate can be induced by TCR activation. And as a result of that activation, you could see a global increase of protein O-GlcNAcylation inside the T cells. And so far, more than 1,000 O-GlcNAc modified proteins has been activated in T cells, and most of them are involved in TCR signaling, metabolism, and transcription. However, T cells does not come in just one flavor, as many of you know that, depending on the types of infection, it can be differentiated to type 1, type 2, type 17, and Treg cells. And of course, there's T follicle helper cells, which is not listed over here. For example, when you facing intracellular bacteria, responsive to the environmental interferon gamma. Those naive T cells can develop in Th1 cells. And on the other hand, when you have helminth infection, instructed by IL-25 and IL-33, etc., those T cells can develop into Th2 cells. And that time, we were very interested into really Treg cells. Those are so-called immune regulatory T cells. By secreting cytokines such as IL-10, TGF-beta can elicit an immune suppressive function, and that are not whose dysfunction is not just associated with autoimmunity, but also its overactivation can lead to reduced tumor immune responses in the tumor settings. And of course, there's a lot of great interest to really study how Treg cells is regulating metabolic responses. So at that time, my former postdoc Bin Liu, teamed up with Oscar in Chris Hallquist's lab to really ask the simple question whether O-GlcNAcylation is really important for Treg cell function or not. So as you probably can expect, that we found that O-GlcNAcylation can also be induced in Treg cells after the activation of TCR, shown over here. So in order to really investigate the function of Treg, OGT in Treg cells, we specifically delete this OGT gene in using FOXP3. So knocking out OGT protein O-GlcNAcylation in Treg cells, leading to a classic scurvy phenotype that's been found similarly when you have germline loss of function of FOXP3 or Treg cells. Those mice died of autoimmune around 4 weeks of age. As you can see that there's enlarge of all the lymphatic organs including spleen and lymph nodes over here. So when you took out these Treg cells out of their bodies, we didn't actually see a difference in terms of number, but interesting enough, we found that the FOXP3 protein itself is actually reduced. So there's a portion of the cells, Treg cells, lose FOXP3 protein expression and become these so-called EX Treg cells that are important for inflammation progression. So as a matter of fact, we found that the FOXP3 protein itself can be O-GlcNAc modified. As you can see over here, if you overexpress OGT inside of the cells, you could see a massive increase of protein O-GlcNAcylation on FOXP3. By the way, this is a FOXP3 IP. On the other hand, when you overexpress the O-GlcNAcase to remove modification, you don't see that band here anymore. So with the help with Dr. L. Burlingame from UCSF, we were able to identify 5 O-GlcNAc modification sites on FOXP3. We mutate them individually or here as a whole. On the protein, we can see that the FOXP3 protein stability is reduced. So as a result, that Treg cell identity cannot be maintained. So in addition to the identity maintenance, we found that O-GlcNAcylation is also required for effective Treg cell function. So as you can see listed here, we use CD44 positive and CD62 negative as a functional marker for effective Treg cells, or KLH1 positive, CD44 positive as effective Treg cell markers. As you can appreciate over here, in both lymph nodes and spleen, these function markers are severely reduced in our knockout mice. And ample amount of studies have really shown that IL-2 signal is not only just important for Treg cell development, but also for the differentiation of the effective Treg cells. So we found that when we treat the mice with IL-2, only the wild type, which are YP negative here, can respond to IL-2 stimuli, showing their increased KLH1 expression, but the knockout cells don't see that induction anymore, suggesting that there's a defective IL-2 response. So when IL-2 adding the cells is actually acting through a JAK and STAT6 pathway, So we actually did RNA sequencing, and indeed we found that the top downregulated genes in these knockout mice are STAT5 pathway genes, and here those are some STAT5 target genes. As you can see, their expression is severely reduced in knockout mice. Interesting enough, STAT5 can be O-GlcNAc modified as well. So we also find the site and mutate it, and compared to the wild-type compartments, these mutant STAT5 cannot efficiently induce target genes such as SOX1 and SOX3 expression in Treg cells. In order to really find out this STAT5 is a functional target of OGT in the Treg cells, we again, with the help from Mike Fra at Center for Immunology at the University of Minnesota, we did a rescue experiment where we crossed our knockout mice to a ROSA26 STAT5 constitutive active mice, where you can only specifically activate STAT5 in FOXP3 and Treg cells. Interesting enough, we can partially rescue the scurvy phenotype. As you can see, their survival rate is extended. More importantly, when you look at FOXP3 protein expression, it can also be partially almost restored compared to the wild-type mice shown in black over here. Not only FOXP3 protein expression, but also effective Treg cell markers, showing here CD44 high and showing here KRIG1 positive cells. The numbers can also be rescued if STAT5 is being overexpressed. Similarly, we could find that these EX Treg cells I just mentioned, those Treg cells lost FOXP3 expression, can be removed when you have STAT6, STAT5 overexpressed. So with that kind of a study, we basically finding that upon TCR activation, OGT can be activated. We don't have a clear answer how that being achieved, but we have some evidence suggesting that calcium signaling is involved. And once this OGT protein is phosphorylated and activated, it can really actually at least the two important proteins for Treg cell function. One is FOXP3. Its glycosylation is really important to maintaining the Treg cell identity. The other one is STAT5. In cooperation with IL-2 signaling, the STAT5 glycosylation is really important for Treg cell suppressive function. I guess, Jean, we will save the questions at the end till the end of the seminar, right?
Yes, we do. So
Okay.
with this opportunity, I'm going to encourage the audience to put in your question to the Q&A box if you want to, as we go along.
Sounds good. Thank you, Jean. So I want to switch gear to really talk about our second story here. So when around the same time when we published this already 3 years ago, we basically demonstrate that O-GlcNAcylation is important for Treg cell function. There's another group showing that actually Treg cells are O-GlcNAcylation also promote Th17 differentiation, and at the same time it has a potential ability to really suppress Th1 differentiation. However, whether O-GlcNAcylation is involved in Th2 responses or not was not known at that moment. As I just introduced at the very beginning, the type 2 immune response is really important for the host to react to helminths, parasitic helminth infections, and also involved in allergy responses. We really didn't directly study Th2 cells. We actually take a different route, really ask the other question. We know that the Th2 activation and requires first order cytokines, including IL-25 and IL-33. But the identification, the source and the activation, secretion of these cytokines is not well known yet. So just one slight background introduction about the parasitic worms. I guess most of you already know that. It's actually still a problem, a worldwide problem nowadays. So this map basically showing that this parasitic worm infection mostly happens in developed regions. Helminths can impair nutritional status in infected people and can lead to morbidity, particularly those heavy infections. And there are medicines, but does not protect against reinfections. And also understanding how our body responding to parasitic worms can help inform us really to understand our response to allergen as well. As I just mentioned earlier, that the response to Helminth infections really utilize two tiers of signaling activation of the immune systems. The Th2 cells and the innate lymphoid site 2 cells secrete those type 2 cytokines to activate a group of cells, including goblet cells, mast cells, and eosinophils, to have these so-called whip and sweep responses to get rid of the helminths from our body. But the upstream helminth sensor and secretion of those cytokines really involves epithelium and IL-25 and IL-33. However, the exact identity of those epithelial cells in not just acting as a sensor but also secreting those cytokines was not known until recently, about 5 years ago. I think one of the reasons for that lack of knowledge in the field is because that compared to micropathogens such as bacteria, viruses, our immune systems using their structural features to recognize that those pattern recognition receptors I just mentioned. However, for allergens and helminths, it's more or less the functional changes, tissue damage that really elicited the immune responses. So it's not harder to really identify these sensors of infection. But fortunately, in 2016, a group of studies identified these so-called tuft cells inside the showing blue illustrated as blue cells over here, are very important for helminth sensing. In homeostatic conditions, you could see very few of these cells, probably one tuft cells per villi crypt structure. But upon helminth infection, you could see massive hyperplasia of these cells, and through studies from several labs, the current knowledge about tuft cells in terms of helminth infection is that somehow they can sensing the infection or the damage to the tissue by helminth, and they secrete a cytokine called IL-25. IL-25 is solely expressed by tuft cells in our body, and this IL-25 can acting on ILC2s and Th2 cells, activate them to secrete cytokines, type 2 cytokines, including IL-14, 13, and 4, to really activate a feed-forward loop to generate more tuft cells and goblet cells. In the end, these type 2 milieu inside the intestine really and activate the so-called whip and sweep response. So at that time, my former postdoc Min Zhao and also visiting scholar Kai Chen Ren, they teamed up together, tried to start to ask ourselves, how do these type 2 cytokines really remodel these intestine epithelial cells? How IL-13 and IL-4 can activate tuft cell differentiation, And also, IL-33 is another alarmins that are important for type 2 immune response. And we don't know where are they secreted in and how they are secreted. So in order to study this, we're basically using a rodent helminth model, which called H. polygyrus here. I will not go to the details how their life cycle is, but we with the help of Jakob Omoti from Washington University, we got those worms and in the lab and then infected the mice. As you can see here, after a few days of infection, when these worms are developed and entering the lumen of the intestine, you could see massive increase of protein O-GlcNAcylation inside of the intestine epithelial cells, and that activation can be sustained. And the time course is very similar to tyrosine phosphorylation with STAT6. I forgot to mention, but STAT6 is the downstream of type 2 cytokines, including IL-13 and IL-4. STAT6 is absolutely required for type 2 immune response in our body. So again, to really, once we found this, activation of protein O-GlcNAcylation, we immediately to ask ourselves what happens if we block protein O-GlcNAcylation. So we generate intestinal epithelial specific OGD knockout. As you can see on the upper left, in a wild type mice, you could after H. pylori infection, you could see a massive increase of these DCLK1 positive tough cells in the small intestine, and that is totally abolished when OGD is knocked out. The quantification is over here. As a result of that, the IL-25 expression is severely reduced. Hence, the animals cannot efficiently expel the helminths. That's a lot of parasitic eggs can be found in the feces. So we also use a different helminth species called Lepostrongylus brasiliensis, and they are they elicited a more stronger type 2 response can be cleared inside of the wild type mice, but we found that this is not the case in the knockout. We know that type 2 immune responses are important for anti-helminth response, and as you can expect, that if you look at Th2 cell numbers and IOC2 proliferation, they are severely impaired in the OGT knockout mice. The type 2 cytokines, including IL-4 and IL-13, are reduced. And then eosinophils and goblet cells, those are basically effector cells to expel helminths. Their numbers and their size are also reduced when OGD is deficient. So with this knowledge, we kind of think we know that OGD is required for this feed-forward loop to be functionally active inside of the animals, but we really don't know when and where this is happening. And also from our previous study, we know that this actually loss of OGT in the intestine can lead to inflammation. So maybe what we found is can have some confounding effectors. So in order to exclude these possibilities, we use a different approach. Here, Dr. Xiaoyou Yang, my postdoc mentor at Yale, kindly provided us this Rusa26 Cree-inducible OGT overexpression mice. So when we cross these Willing Cree to only express OGT-specific intestinal epithelial cells, in those mice don't have any inflammation phenotypes. As a matter of fact, if you look at their tough cells, both at the naive state and after inflammation, tough cell numbers are increased. We also took the opposite approach to overexpress the O-GlcNAcase inside of the intestine epithelial cells. We could see the opposite, where these tough cell markers such as DCLK1 and POU2F3 are reduced. And then the next question we ask whether this OGD is inside the progenitor cells or in the mature cells is really important. So one thing we did is that in collaboration with Dr. Haibin at Xinjiang Medical University, we specifically knock out the OGT in a Cre-ER-dependent manner in OLFM4 progenitor cells, and we could see that reduced DCLK1 expression after OGT knockout. However, on the other hand, when we using DCLK1 Cre to specifically knock out OGD in tough cells, we don't see any defect in the numbers or anti-helminth response. So those data are suggesting us that OGT is acting inside of the progenitor cells that are important for the differentiation of tough cells, but not so much in the mature tough cells. Because STAT6 is absolutely required for these anti-helminth responses, so we ask us whether OGT is regulating STAT6 pathway. Indeed, that's the case. First of all, if you're treating intestinal organoids with OGD inhibitors, showing over here, we use two structurally different inhibitors, both of which can suppress tyrosine phosphorylation of STAT6 after IL-4 treatment. In vivo, when we isolate the intestinal epithelials out from the knockout mice, the tyrosine phosphorylation is reduced compared to their wild-type mates. As a matter of fact, that STAT6 itself can be O-GlcNAc modified. As you can see here, OGT overexpression increase glycosylation levels of STAT6, and at the same time, increase tyrosine phosphorylation of STAT6. We also look at STAT6 transcription activity using luciferase, showing over here that only the wild-type but not the catalytically dead OGT can increase the STAT6 luciferase activity. We also using OMSI and the inhibitor of OGD and found that the IL-4 induced STAT6 transcriptional activity is reduced when OGD is inhibited. So again, we ask help from our collaborator Al Bellingham at UCSF. We just sent him purified STAT6. Their group did the mass spec to identify O-GlcNAc sites, and very gratifying that we found 3 clusters of O-GlcNAcylation in the C terminal of STAT6. One cluster is very close to the tyrosine phosphorylation site. The other 2 are actually in the transactivation domain of the STAT6. So we made individual mutations or in combination and found that mutating either of them going to reduce the modification on STAT6. And particularly these 2 clusters in the transactivation domain is a major site, basically showing over here, mutating this transactivation domain almost remove all the modifications on STAT6. As a result of this reduction of O-GlcNAcylation, we could see a reduced luciferase activity to a certain extent at the baseline, but most importantly after IL-4 treatment. We also ask ourselves whether that modification is really regulating tough cell development. So we eyes on this POU12-3 gene, which is known as a tough cell specific transcription factor, and we found that wild-type, but not the glycosylation mutant STAT6 can activate POU12-3 transcription. This is not just in the cell culture systems, but also in intestinal organoids. If we transduce these mutant STAT6 into the organoids, their ability to increase tough cell markers was impaired. So all this is good, and to really we always want interesting to use genetic model systems approaches to test our hypothesis. Again, we collaboration with Haibin and also with the help from Gen Pharmatech, we generate this vinicry specific constitutive active STAT6 mice, where these STAT6 constitutive active STAT6 only express Weening tree. As you can see here, the tyrosine STAT6 is massively increased in those transgenic mice. And we crossed these transgenic mice to O-GlcNAc knockout mice, and we are gratified to really see that the loss of function of the top cells showing here DCL staining, and also loss of goblet cells showing by L-cyan blue staining can be at least partially rescued by just overexpressing STAT6 in the intestine. So from these data, we have our model that we think these OGT in the intestine progenitor cells can glycosylate and activate STAT6, and that activation of STAT6 is important for the transcription of POU12-3, which drives the differentiation of top cells. Top cells secrete IL-25, activate ILC2s and THC2s, and these type 2 cytokines acting again on STAT6 to form this feed-forward loop that is required for these so-called whip and sweep responses. But we didn't want to stop there, because we asked ourselves, it appears that top cells itself cannot expand all the phenotypes we see. And also at the same time, we don't know where this IL-33 can be secreted and can be regulated. So we did RNA sequencing of these OGT knockout versus wild-type mice, and found, interesting enough, those most downregulated genes called GSDMC2, GSDMC3, and GSDMC4. On the other hand, if you treat the organoids, intestinal organoids, with IL-4, and these GSDMC genes are most abundantly massively overexpressed after treatment. We were fascinated by these findings, and of course, as many of you know, this GSDMA family, there's GSDMA A, B, C, D, and E in our body, and the current study showing that all these GSDMA proteins has these pore-forming N-terminal once it's activated by caspase-3 and forming pores on cell membrane. Probably the most studied is GSDMD in macrophages, as listed over here. These GSDMD proteins can be activated through these canonical inflammasome pathways, can also activated by these non-canonical inflammasome pathways. The activation of GSDMC can lead to the cleavage and then pore formation on the cell membrane. And that pore formation can has twofold function. First, it can mediate the secretion of IL-1 beta and IL-1 those type 1, these IL-1 family of proteins. Those proteins lack signaling peptides, so they has to rely on these non-conventional ways to secrete it out. If these pores cannot be repaired and they forms a lot on the cell membrane, these eventually leads to this specialized cell death pathway called paraptosis. Once we found this GSDMC, we look at all these GSDMA families and found only the GSDMC genes can be induced after helminth infection. This expression is also much higher compared to A and D and E in the intestine. Mostly exciting finding is that after helminth infection, not just the protein expression, but also the cleavage of the GSDMC to forming pores can be induced by H. pylori infection. Also, to be consistent with our RNA sequence data, organoid studies from wild-type versus knockout mice found that their expression is absolutely requires STAT6. Without STAT6, GSDMC cannot be induced by IL-4. Again, we used luciferase assay and found that OGT can increase the expression and the transcription of GSDMC here, inhibiting OGT by chemical OSMI or overexpression OGA can reduce the expression. We know that this STAT6 glycation is important because this mutant STAT6 cannot efficiently induce GSDMC transcription activity or gene expression inside the organoids. Once we have these findings, I immediately talked to Sean Gao, my graduate mentor and also the founder of the GEM Pharmatech. And I know he has been working on all these gastamine families, and I show these preliminary data, and he was generous enough to show us these gastamin C floxed mice. And then we get these mice to University of Minnesota, cross them with William-Cree, and infected them with helminths. And we were excited that time to find that after infection, these gastamin C knockout mice has reduced tough cell marker expressions. Goblet cells was also reduced over here. As a result of that, there's more helminth eggs can be found in the feces. One thing you probably can see is that the decrease of these tough cell numbers or increase of the eggs was not as dramatic as the O-GlcNAc knockout, suggesting that gastamin C is only one pathways that are mediating the effect of O-GlcNAc. But we are excited to find that at least the gastamin C are partially important for anti-helminth responses. However, it took us some time to really figure out how this GSDMC works. When we using organoid cultures and treat them with IL-4 or without, we found there's no difference at all of these tough cell markers or any other genes related to helminth infection. Here, just another example showing that basically no much of gene expression had been changed between the two types. So at that time, we think, okay, maybe this gastamine function in a non-cell autonomous pathway. So as I just mentioned in macrophages, this gastam D can, once activated, forming pore on the cell membrane, it is going to mediate the secretion of IL-1 beta and IL-18 and cytokine secretion. So maybe inside of the intestinal epithelial cells, this gastam C can have the similar function, but instead they're going to mediate the secretion of IL-33, which is also IL-1 beta family, which also lacks the signal peptides. So first we did some staining to really confirm that, okay, intestinal epithelial cells can express IL-33. So we confirmed this. The specific antibody of IL-33 was determined by knockout mice. As you can see that in addition to the lamina propria cells, some of these intestinal epithelial cells can express IL-33 at a very high level as well. And those on the crypt regions of the intestine are actually goblet cells. We co-staining with goblet cell markers such as AG2 here. When you look at the gastamin C at the RNA level, all these gastamin C genes are mostly expressed by these so-called transient amplifying cells. Those are progenitor cells of the intestine. However, if you do the protein expression, on the other hand, you could see that their expression is mostly happening in the villus crypt, in the villus of the intestine. And particularly, most important, after H. pylori infection, you could clearly see their membrane location of the gastamin C. To test whether gastamin C mediating IL-33 secretion, so we did this co-expression and found that IL-33 secretion into the medium can be mediated by gastamin C overexpression. Most importantly, if we do getting this intestinal epithelial out from the knockout mice and look at IL-33 secretion into the medium, and either by Western or by ELISA, you could see that their IL-33 secretion was severely impaired. And to demonstrate that's important, we really giving back IL-33 by injection into these knockout mice and found that the defect in helminth response can be rescued once IL-33 was injected into these mice. I think this will be my final slides of the data. So again, I have long-term interaction with all these former trainings of Dr. Gao. Here is Zhao Yuning. He is also at Nanjing University. So we simply ask ourselves whether GSDMC is only important for helminth infection. What about other conditions, for example, intestinal inflammation? So we did two simple experiments. First is using IL-10 knockout mice. We know that this is a colitis, spontaneous colitis model. Interesting enough, we found the GSDMC protein can be induced, and also its cleavage can be induced in these colitis models. When we do a double knockout, knockout GSDMC on the background of IL-10, we could find their colitis is improved. We also look at this DSS chemical colitis. We found that the GSDMC protein can be induced transiently, but most important, if we are using helminth infection to induce GSDMC expression after DSS colitis, and we could see that the knockout mice over here are doing better in terms of their response, suggesting that loss of GSDMC can alleviate some of these colitis damages inside of the cells, inside of the mice. So this is basically my summary slides for the second story. Basically, the punchline is that we found that inside the intestine epithelial cells, OGT protein O-GlcNAcylation is responsive to helminth, which is gonna activate the STAT6. The activation of STAT6 can do two parallel activities for type 2 cytokines. First, it can induce the differentiation of type cells and IL-23 secretion through POU2F3 transcription. Second, it can increase IL-33 secretion from cells such as goblet cells in the intestine via activation, the transcription of GSDMC. So together, these type 2 cytokine milieu is important for the WEEP and SWEEP responses. So with that, I just want to thank all my current and former members. I think along the way I have introduced their contributions. I also like to thank all my collaborators, particularly Dr. Shiven Xianggao and Zhao Yuling. And also I would like to thank O-GlcNAc for their mass spec expertise. Without their help, we couldn't really be at this stage. We also, of course, I want to thank my findings from NIH, ADA, and University of Minnesota. With that, I like, thank you for your attention. I'd like to take any questions that you might have.
Well, thank you so much, Haibin. It's a very dense presentation with packed data. Appreciate, really love the science. There's a lot of knowledge to learn. It is a big area for the future development. So I do see questions in the Q&A boxes. I'll read it out to you. So the first question: did OGT deficient Tregs lose FOXP3 due to FOXP3 protein degradation? Is it related to other post-translational modifications of FOXP3, such as phosphorylation or ubiquitination?
Oh, this is a great question. Yes, the short answer is yes. I didn't have time to include the data, but basically what we found is that glycosylation is stabilizing FOXP3 protein. Without a glycosylation, the FOXP3 protein ubiquitination gonna be increased, and you're gonna have faster degradation of that FOXP3. So yes, at least so far we know that this glycosylation can suppress ubiquitination on FOXP3 and stabilize it. we don't know whether there's a crosstalk with phosphorylation yet, and we don't know how exactly glycylation talks to ubiquitination on phosphatase protein yet. Those are great questions.
Great. And the second one, how does STAT6 glycosylation promote its phosphorylation? Does STAT6 O-GlcNAc facilitate its recruitment to JAK?
Oh, that's another great question. Okay. So to be clear, we found that when we treat the cells with OGT inhibitors or knock out OGT, there's reduced tyrosine phosphorylation of STAT6. On the other hand, if we overexpress OGT, there will be increased tyrosine phosphorylation. However, when we mutate glycylation sites on STAT6, we found that tyrosine phosphorylation is not affected by those modifications. So the conclusion from this study is showing that the glycylation can, in addition to acting on STAT6 directly, can also maybe target other proteins such as JAK to phosphorylate STAT6. We actually have evidence that JAK can be O-GlcNAc modified as well. We have not studied that yet in terms of the activation of STAT6. But at least that's possibility there. So we think the tyrosine phosphorylation of STAT6 is independent on glycylation on STAT6 itself, but rather the glycylation on STAT6 is really can actually control serine phosphorylation on STAT6, which I didn't have time to show. There are some inhibitory serine phosphorylations on STAT6, and we found that glycylation can suppress this serine phosphorylation on STAT6. I hope that answered your question. Again, these are great questions.
Thank you, Haibin. I think we probably have time for one more question. I'm going to read it to you. It's also about the crosstalks between these signaling pathways, but this is more on the structural or 3D positioning. So phosphorylation and the glycosylation, you showed some of the evidence where it's happening on serine threonine, which are the same amino acids on the same protein. So structurally, are these competing? Are these modifications competing the space? If you think about the structure of the protein after the modifications.
Yes. The short answer is yes, but not just that. So there's a lot of so-called yin-yang sites on these proteins, meaning those serine and threonine can both O-GlcNAc modified and phosphorylated. And when that happens, so of course there's a competition between these two modifications. You can either be O-GlcNAc modified or either be phosphorylated. And in addition, there are a lot of O-GlcNAc sites are very adjacent to the serine phosphorylation sites, and in that case, the interplay between these two modifications is actually context dependent. They can compete with each other as well. They can also really has synergistic effect. It really depends on the site and the protein. And you have to study this case by case.
I see. Great. Well, thank you for the answer. I think we're very, very close to the top of the hour. I'm checking one more time in the Q&A box. I think that's all the questions from the audience today. And one more time, thank you, Haibin, and thank you everybody for joining us today. We look forward to seeing you again in September.
Oh, thank you for GM Pharmatech. As you can see, that my research is heavily animal dependent, rely on animal models, and as I alluded, I have help from many scientists, including GM Pharmatech, to providing us these models, and I'm very appreciative of that. And we'll continue using your service, and we at least have one mice on the pipeline right now. So I look forward to our collaborations, and I hope to come back in the near future to talk about our new findings. Thank you,
Haibin. Yeah, looking forward to that. Thank you, everyone. See you next time.
Bye.
Bye-bye.